Today we are going to study about the assort and biodegradation of xenobiotic compound. In this lecture we will discuss about meaning of xenobiotic compound, types of recalcitrant, the assort of xenobiotic compound, general features of biodegradation, biodegradation of halogenated compound, and the origin of capacity to degrade xenobiotics. In the first slide we will see the meaning of xenobiotic compound. Xenobiotic compounds are man-made chemicals that are present in the environment unnaturally high concentration. The xenobiotic compounds are either not produced naturally or are produced at much lower concentration than man. Normally, the microorganisms have the capacity to degrade all naturally occurring compounds, are also able to degrade many of the xenobiotic compounds, but they are unable to degrade many others. The compounds that resist biodegradation and thereby persist in the environment are called as recalcitrant. Next, the factor causing molecular recalcitrant. The xenobiotic compound may be recalcitrant due to following reason. Number 1. Xenobiotic compounds are not recognized as substrate by the exhaustive degradative enzyme. They are highly stable, that means chemically and biologically inert due to the presence of substitution group like halogen, nitrosulfonate, amino, methyl and carbamyl group. They are insoluble in water or are absorbed to external material like soil. They are highly toxic or give rise to toxic product due to microbial activity. Their large molecular size may prevent the entry of microbial cell, inability of the compound to induce the synthesis of degrading enzyme, and the last one is the lack of permease, which is needed for the transport into the microbial cell. Then types of recalcitrant xenobiotic compound. The recalcitrant xenobiotic compounds can be grouped into 6 types. One is halocarbon, second one is polychlorinated biphenyl, third one is synthetic polymer, fourth one is alkyl benzene sulfonate, fifth one oil mixture, and sixth one other compound. The structural features that make the xenobiotic compound that are resistant to microbial degradation include the presence of halogen in the place of hydrogen in the molecule. The carbon halogen bond is highly stable and its cleavage require considerable energy. Second feature, substitution of hydrogen by other group like nitro, sulfonate, methoxy, amino and carbamyl group. Third one, cyclic structure such as aromatic compound, cycloalkane and hydrocyclic compound are more recalcitrant than linear chain or aliphatic compound. And the last one is the branched linear chain which are resistant to biodegradation. In general, the more complex is the structure of xenobiotic compound and it is more resistant to biodegradation. Let us see halocarbon. This compound contains different number of halogen atom in the place of hydrogen atom. They are used as solvent, propellant, condenser unit, insecticide and herbicides. Second one, polychlorinated biphenyl, shortly called as PCB. These compounds have two covalently linked benzene ring having halogen substituting for hydrogen. These PCB are used as plasticizer, insulator coolant in transformer and as heat exchange fluid. They are both biologically and chemically inert to various degree, which increases with the number of chlorine atom present in the molecule. The recalcitrant nature of the above two compounds such as halocarbon and PCB are due to their halogenation as well as their cyclic structure. Third one, synthetic polymer. These compounds are produced as plastic, example polyethylene, polystyrene, polyvinyl chloride and nylon, which are used as a garment and wrapping material. They are recalcitrant mainly due to their insolubility in water and molecular size. Fourth one, alkyl benzene sulfonate. These are surface active detergent superior to soap. In this, the sulfonate group present at the one end can resist to microbial degradation while the other end become recalcitrant if it is branched. Fifth one, oil mixture. Oil is a natural product, has many components and also biodegradable. Oil is recalcitrant mainly because of its insolubility in water and due to the toxicity of some of its components. The other xenobiotic compounds include many pesticides which are based on aliphatic cycling ring structure that containing substitution of nitro, sulfonate, methoxy, amino and carbonyl group. In addition to that, they also containing halogens. This substitution make the compound to recalcitrant. Next, what are the hazards from xenobiotic compound? The xenobiotic present a number of potential hazards to human and the environment, in which number one, toxicity. Many xenobiotic like halogenated and aromatic hydrocarbons are toxic to bacteria, lower eukaryotes and even human beings. At low concentration, they may cause various skin problems and reduce reproductive potential. Second one, carcinogenicity. Certain halogenated hydrocarbons have the ability to produce cancer. Also, many xenobiotic including DDT and PCB are recalcitrant. As a consequence, they can show bioaccumulation or biomagnification. The biomagnification occurs mainly because of two reasons. Once these compounds are continuously taken from the environment and accumulated in the lipid deposit of body, for example, 100-fold accumulation of DDT by plankton from water. Then this organism can be consumed by the other organism in a sequential manner. For example, the plankton is eaten by small fish, the small fish eaten by large fish, and the large fish can be eaten by sea eagle. And the last one is the xenobiotic compounds are produced and used in large quantities, which favors their accumulation in nature. Then we will see the biodegradation of xenobiotics. In this, the degradation of alkanes and aromatic hydrocarbon generally occurs as follows. Initially, an oxygenase first introduces a hydroxyl group to make the compound reactive. Then the hydroxyl group is oxidized to a carboxyl group. After that, the ring structure is opened up. Then the linear molecule is degraded by beta oxidation in order to yield acetyl-CoA. Next, hydrocarbon degradation. In this, halomethanes. Halomethanes are transferred into methanol by the enzyme methane monooxygenase. This enzyme is commonly occur in a number of methyl tropes. Then this reaction is anaerobic and uses oxygen derived from water. In this, methanol is oxidized to carbon dioxide and water through formaldehyde and formic acid. Second one, cyanide. Cyanide is toxic to biological system and even microorganism. Therefore, the disposal of cyanide is strictly controlled. The cyanide present in the soil is degraded by many of the bacteria and fungi. In this process, the cyanide is converted into carbon dioxide and amino group by the action of bacterial enzyme. The third one, aliphatic hydrocarbon. The aliphatic hydrocarbon may be saturated or unsaturated. Compared to unsaturated, the saturated aliphatic are easier to degrade. The biodegradation of N-alkanes is catalyzed by oxygenase to produce carboxylic acid, which is then degraded by beta oxidation. The oxidation may involve the methyl group at the one end of N-alkane, or it may occur at a beta methylene group. In sometimes, both terminal methyl groups are oxidized to yield a dicarboxylic acid. This reaction is used by many microorganisms for the degradation of branched chain L-alkanes. Next, aromatic hydrocarbon. These are oxidized by dioxygenase to catechol, which is further metabolized by two separate pathways. In case of ortho ring cleavage pathway, the dioxygenase cleave the ring between the two adjacent hydroxyl group and sequential catabolism of the product succinate and acetyl-CoA. Alternatively, the enzyme 2,3-dioxygenase cleave the ring between the carbon atom and having OH group and an adjacent carbon lacking an OH group. The product at the end of sequence reaction are acetaldehyde and pyruvate. Next, biodegradation of halogenated compound. The biodegradation of halogenated compound involves two steps: elimination of the halogen group and degradation of the non-halogenated product molecule. Removal of halogen molecule may occur either directly involving the removal of hydrogen halide, or it may be involved the substitution of halogen by H, OH or thio group. Then the direct halogen removal produces a double bond. The mechanism involving a allergen substitution, especially by OH. The aerobic degradation of halogenated aromatic compound usually involve the following step. Number 1. Addition of OH group by a deoxygenase to yield chlorinated catechol. In the second step, the cleavage of ring by ortho or meta-cleavage often result in the halogen but being eliminated from the aliphatic product, which may lead to an accumulation of toxic metabolites. Next, cleavage of the ring by ortho or meta-cleavage, then elimination of the halogen from the straight chain product, and finally degradation of the aliphatic hydrocarbon so produced. Next, we will see the origin capacity to degrade xenobiotic. Generally, the continuous exposure of microorganism to xenobiotic compound can often lead to the evaluation of metabolic process needed to wholly or partially degrade the xenobiotic compound which are present in the environment. This capability may arise due to mutation and transfer of plasmid bone gene. Gene mutation occurs spontaneously at a low frequency, but the short generation time and very large population of microorganisms make mutation a potent source of genetic variation. Mutation can be expected to either modify the active site of an enzyme so that it has an increased affinity for the xenobiotic, or it can be eliminate regulatory control and enhance its production. These changes only enhance the rate of degradation of a xenobiotic and rarely generate a new enzyme function. With the help of plasmid transfer, the microorganism will acquire a new enzyme activity which are involved in the xenobiotic metabolism. This type of plasmids are normally found in some gram-negative bacteria, for example Pseudomonas. Then the plasmid transfer allow a microorganism to acquire the gene which are needed to complete the pathway for a xenobiotic metabolism and to gain the gene which improves the rate and the nature of the degradation. For example, when a mixture of alkalogen and Pseudomonas strain were maintained in laboratory, the alkalogen species acquired the plasmid and the ability for ortho-ring cleavage from Pseudomonas strain. This is a good example of production a natural recombinant species. Nowadays, more attempts are being made to develop genetically engineered bacteria having recombinant pathway for xenobiotic compound degradation. Next, we will see how the genetically modified microorganisms are helpful in bioremediation. Normally, the accumulated toxic and persistent compounds, especially the xenobiotic compound present in the environment, cannot be degraded efficiently by the natural metabolic diversity of the autotomous microbes. The novel nature of this pollutant, which include unusual chemical bonding or substitution with halogen or other functional groups, that makes them resistant to natural degradation by the inhabiting microorganism, and due to the lack of appropriate metabolic pathways. The hydrophobic nature of various toxic contaminants in environment such as toluene, organochloride and polyaromatic hydrocarbon accumulates in the microbial cell and disturb the cell wall. Then the prolonged persistence of this compound is due to the inefficient transport system and their limited bioavailability because of their hydrophobic nature. However, the natural inhabiting consortia at various physiological environmental conditions will provide the genetic sources of novel metabolic capabilities and the possibility of degrading these recalcitrant chemicals using genetically modified bacterial strain through the introduction of different enzyme activities from a number of bacteria into a desired microorganism. During the long evolutionary process, different microorganisms gain unique abilities to degrade persistent compound at contaminated site by the physiochemical pressure in those environmental conditions. The potential of this natural microflora can be utilized by identifying and engineering catabolic genes. Then the use of indigenous microflora instead of toxic strain is always naturally advantageous for the construction of recombinant microorganism, because the indigenous microorganisms are more prone to interact with total population and can withstand to the complex stressful environmental conditions. For example, catechol 1,2-dioxinase, toluene dioxinase iron sulfur protein component, benzene dioxinase, naphthalene dioxinase, dioxine dioxinase of Spingomona species, and many ring activating dioxinase. These are some example of novel enzyme identified for recombinant DNA technology in order to improve the strain for better bioremediation capabilities. The successful application of genetically modified organism for bioremediation is based upon the successful establishment of the engineered microorganism in the environmental conditions and after the completion of the desired objectives. There should be an appropriate mechanism for their removal from the site of action. Then the potential risk associated with the release of genetically engineered microorganism into the environment has originated the necessity to construct the biological contaminant system by which bacteria are killed in a controlled suicide process. The strategies used for engineering any microbial cell capable of degrading specific compound and also to make it able to remain viable for many generations are very different from the technique used to develop recombinant strain for protein overproduction. A balance between organism growth and energy consumption for the macromolecule synthesis, capability of the genetic vector to carry the large foreign DNA, then ability of the cell to replicate the DNA exactly, and also maintenance of the plasmid by the cell are some of the issues which must be kept in mind during the designing of any strategy. To develop an efficient microorganism, the number of expression system containing various promoters and the regulators has been discovered by different groups. The modification of enzyme affinity and specificity, bioprocess development, pathway construction and regulation, bioactivity, bioreporter sensor development for chemical sensing, endpoint analysis, and toxicity reduction are the main approaches to develop genetically engineered microorganism. The general mechanism used for modifying a microorganism or an enzyme by rational modification are rational designing, directed evaluation, saturation mutagenesis, and metabolic engineering. The rational designing involved the construction of a single microorganism having the assembly of desirable biodegradation pathways or enzymes from different organisms in order to perform specific reactions using recombinant DNA technology. Directed evaluation technology involved the engineering of the enzymes with desired characteristic using site-directed mutagenesis. Then in saturation mutagenesis, the engineered catalyst having mutation at key sites are developed. The key sites are targeted for interactive cycles of mutagenesis until the gene for desired level of engagement in the gene property is achieved. In the last case, that means the metabolic engineering, the process of the engagement in the production of a specific cellular component by the optimization of genetic and regulatory means. The growing awareness about the impact of environmental pollution has triggered a number of global remediation actions in recent years. In order to apply a technique to mitigate the effect of a pollutant on the environment, the first requirement is to have a fair idea about the presence of a particular target pollutant. Although there are numerous traditional methods for pollutant monitoring, for example, to monitor heavy metals such as cadmium, zinc, lead, arsenic, and copper present in the polluted site. And also, recently, several developments in the field of recombinant DNA technologies, such as the development of suicidal genetically engineered microorganisms, have also been carried out to achieve safe and efficient bioremediation of the contaminated site. These are some advancements in genetically modified microorganisms which are used in bioremediation process. However, Some anticipated risks are found in genetically modified organisms. First, it should be noted that from the biosafety viewpoint, not all naturally occurring soil bacteria are ideal as bioremediation agent. For example, Burkholderia cepacia as potential as an agent for bioremediation and for biological control of phytopathogen. However, it is a human pathogen known to be involved in cystic fibrosis and it is resistant to multiple antibiotics. The transgene is usually derived from another soil microorganism. Thus, no new gene is added to the soil microbial community. It is also very probable that the introduced engineered strain will not survive for long in the soil environment, at least not long after its specific substrate is exhausted. However, several cutting-edge strategies have developed for mitigation of probably risk of genetically modified organisms to achieve efficient and safer bioremediation of contaminated site. The application of molecular biology based technique in bioremediation is being highly used and has provided useful information for improving of bioremediation strategies. Furthermore, environmental metagenic data from soil and sea can be useful source of genes. Then the combination approaches such as the genome shuffling are also useful for generating new genes or modifying enzyme activities in order to allow efficient bioremediation. Thank you.